Cool. Yeah, that's all I had. Questions? We got about 7 or 8 minutes, and then everybody can go on to the next thing. Christine, are we doing a microphone? Are we doing any questions?
Yeah. So it looks like Kenda has some questions. Are you ready to ask those?
Yeah, I'm ready to go. First of all, Colin, Colin, great talk. I love this, and you're not the only bunch of net jobs out there. There's a lot of us doing biological RSRU. In fact, I'm putting something in the link that I'd like everybody to look at. We have started a collaborative to talk about this. So thank you. I love this talk. We have started a collaborative. We want everybody to join. We're kicking off right now. We're actually interfacing with LSIC. So enough of my blurb, my question and my interest. So one of the things that we're doing, I love the natural perspective. And my question is, when you guys look at your redox lever in the wetlands, have you thought about including perchlorate reduction within that redox tower? Because that kind of fits in there between oxygen and nitrate reduction. And even though it can get limited by oxygen utilization, it can fit in there between nitrate utilization, and sometimes, invokes at the same time. So I'm just very curious if you guys have tried to look at how that would impact your system, having that naturally get utilized in your system, because that's something that we actually have an experiment running doing that right now with Martian regolith, using a natural system.
I have not. Thank you very much. And that was actually news to me then, because if we going back to that the big really dense slide there. I was looking at perchlorate reduction for oxygen removal, and then going to the chloralkali electrolysis in order to produce a couple different products. So I guess hydrochloric acid, and I think it was caustic soda or something, or sodium. Yeah, it was sodium hydroxide.
Okay.
And that's about as far as I got with it, because in the mining field around here, I mean, I can only speak from personal experience and the things that I have seen. I've never had to mess around with that. So it just hasn't even been on my radar, other than I know it's a real potential for being useful, but then you're dealing with, miscible salts after. I guess that's kind of you get the idea. It's like, what do you do? How do you pull the salts then, that chloride ions, out after the fact?
So we're figuring out, that's part of our experiment, and we got NASA. It's part of our experiment. So you and I, we need to talk, because we're trying to figure out, those are some of the questions we're trying to answer. And Patrick, to answer your question, just like what Colin's saying, we actually took a natural self-organizing perspective, and we're actually using samples from a natural perchlorate reducing environment that's also a Mars analog from my dissertation research. So they're an ecosystem that we're using to see if they can survive and do their thing and reduce the perchlorate, and also start to help build our simulants into a pre-soil product. That's one of our experiment we're actually running right now. So I would really love, Colin, to sit down and talk with you guys a lot about this and seeing what you're doing with your wetlands versus, because ours is from a subsurface groundwater supported ecosystem versus an open wetland. So it would be really interesting to kind of discuss that.
Yeah, there's the benefit that, I mean, if you can go ahead and like a heap leach process and pull it all out of the ground, instead of having to do it in situ. We're still doing it kind of in situ, but not really. I mean, once it's out of the pile, technically, I guess that's the definition of in situ, is really doing it truly initially in place. Like if you're thinking about permeable or non-permeable reactive barriers, or anything that's going on actually in the ground, versus bringing it out where then you can manipulate more easily those different constituents. Whether just a lot of times it's just putting a lid on the darn thing and sealing it up, and then just letting it go through its own system. So yeah, please.
Yeah. Let's definitely talk, because right now we're doing it lab based. So we have a anaerobic and aerobic treatment. We're doing it a couple different ways, and so we're controlling things laboratory wise. But I'd really love for us to talk, because I think there's a lot of crossover on what you guys are doing and what we're trying to do with this experiment too. So I just found this really interesting, and thank you so much for doing this work. This is super exciting and exactly where we need to be.
Sure. And Michaela, she had just graduated. She's still on here. If You need people. I can't afford her. No, I'm serious. I can't afford her. She's, I'm just happy that she was able to help me through these current papers that we're starting to work on. But yeah, if you need somebody, that's your person right there. Yeah, do we? Thank you. Then, Kenda. Yeah, thank you very much. Love to talk. Do we? I think we've only got a couple minutes left here.
Yeah,
Are there
go
any
ahead. Somebody
other?
else. Richard's got a question. Sorry, I will shut up now. Thanks. Thank you.
Oh, thank you. No, seriously, thank you.
Yeah, go ahead, Richard Parker.
Hello. Hi. Excellent talk. I really enjoyed it. Not really familiar in that area, but I could definitely see both the terrestrial and extraterrestrial applications. So thinking of the hypoxic like bioreactor, which was like quite prominent in your talk, that's really prevalent to spaceflight because of the naturally hypoxic nature of microgravity. How might you imagine the like methanotrophic bacterial community changing in a hypoxic environment? How would you imagine that would help them adapt to that stress? Like if you saw that in your bioreactors and you started seeing a succession in the community, how would you interpret that, and how would you adapt that? And does that make sense? I'm trying to keep it open-ended. There's a series of 3 questions there to help try and structure your thinking based on like that redox ladder that seems to be really important. And so do you go for it?
Yeah, and I think it makes things easier. Boy, I don't want to get myself into starting to just kind of going off the cuff. I don't have a good answer for you. I think that's maybe the best answer in the world for right now. I don't have any personal experience with actually producing methane. Now, there are others that I speak to in the field who do. They're usually going straight to methane in batch reactors. Now, as you'd mentioned, and I guess that hadn't occurred to me how much more difficult it is to keep something aerated versus allowing it to go dancing through those redox steps as the different electron acceptors and donors are burned up and used up earlier within a treatment train, assuming that hypothetical oxidative, and then on through the hypoxic and then all the way down to methanogenic. Good question. I guess maybe I'd have to do a little bit more reading just for a little bit better context to get you a better answer. But I don't want to stick my foot in it, so to speak. But that is a concern because in the is Dr. Mark Weisvogel? Frank, did I get that name right? If you can give me a note. Okay, yeah, we're the guy with the space coffee mug. He is someone that we're trying to talk to here next, because my concerns are how do we keep it aerobic? How do we keep a system moving? And then, what will that, say if we're on the ISS, what does a treatment system like this look like? Is it soft? Is it hard shelled structures? Because all of those different, how are you introducing the oxygen? Because in a lot of ways, the different studies that have been done on flame in space, producing those beautiful little glowing bubbles with this aurora on the outside of these light colors, and you can that determining the ability of the system to burn, or of that candle wick to burn, for that oxygen being able to get to the inside of it. I see those as being, I'm not sure if this is really answering your question at all, but like those, as far as design components, I think there's some corollaries there that we can use as designers to make a system that, because like you said, it's more difficult, I think, in this case, to get the O2 into it than it is to allow for the redox, the self-organization to occur. And I apologize if that didn't answer your question. You just gave me something to think about, though. So I was kind of off the cuff a little bit.
That was really good.
That
was really good. Thanks. I like the way you came up with like a hardware response, aka like a sewing oxygenation system that would be needed for space flight. And I just had one other little follow-on question. Was it a coincidence that you had 8 different reaction chambers, and there appears to be about 8 different redox levels in the redox ladder? And do you
Sure.
like consider augmenting the different stages to promote different types of reactions and then monitor like the microbial communities, or is that future research? Am I over? Well, thanks again.
Oh, sure, sure. Yeah, and that particular system, those top 8 boxes are only used for the removal of aluminum. And I was up there a couple weeks ago during the last thaw, and they were running 170 gallons a minute, which is really fast. That's the point where they're actually starting to spill over the sides of those units. And the thing to remember is that because if you think about that old video game called Cubert, where you got that little guy with the big nose and the 2 legs, and he's jumping down on those different stages. Anybody? Yeah, I see Christina's like, yeah, I remember that back in the day. That was, the video game galleries of the sounds of our youth when you're anything over 40, I guess nowadays. But Cubert had to jump down between those different stages. So for a flow-through system to maintain its rate without spilling over the sides, but then still be able to remediate everything that is biologically remediatable, so to speak. I mean, because once again, salts, no. Sodium chloride, no. Nutrient salts, yes. That's a whole other talk, though. But you have to keep in mind that if you're going to have 10 gallons a minute going through a system that's aerobic, if you have by the next step down and you're going into nitrogen, it's less efficient. So you're either going to create a longer series of units, there will be more, or you got to split them into parallel, just like batteries. And then you get to the next redox step, you got to have another split. And then the next one, another split, or they're just longer, a longer treatment train. So you could essentially go 1 to, say 100, where all the aerobic remediation can be done in the first 2 boxes. And then by the time you get down to methanogenesis, just, for a hypothetical, it might take the last 30. I'm just pulling numbers out here, but in general, and you just go back to the equations of, okay, if methane or CO2 reduction will yield 4 ATP versus aerobic, which is 32, you got an 8-to-1. And so you just look at the ratio of what is the starting point, what's that most efficient system, and then you determine then the overall volume of the amount of units or their size going downstream, so to speak, and then all of our units, and those models that I showed you, the black boxes there, they're dated now, even for where we are right now, for our systems. So you can build them either larger, split them into parallel series. There's another one, but those are kind of the design parameters that you need to allow for a flow through system that is that it's gravity flow. It's technically passive without water spilling over the sides. So that's just something to keep in mind for designers. And that's mostly what our paper that we're getting ready to publish is about. Here, if the self-organizing wetland bioreactor, both as a tool and a means to further science and do actual scientific research. And most of those boxes there, the seals on those lids, not good. They weren't good enough for what I wanted. I was hoping I was going to be able to get reductive there. Now they will go reductive when it's slow, because I could smell hydrogen sulfide coming off of the first unit when it's full of aluminum hydroxide. So the waste product that was coming out in that water was sucking up all the oxygen and providing so much surface area that that unit particularly did go reductive and started reducing sulfate, which is kind of what we wanted. But it's basically, whether you seal the thing or not. Now, of course, you have to have, some means to be able to allow, pressure off gassing, so the thing won't burp itself, and it allow for, because otherwise you'll get like an air pocket, which can slow flow down, so you won't get a full flow through, and you may lose 2/3 or something like that of your flow through capacity. So there's still, you got to be able to link the atmospheres between them, but such that the overall atmosphere is not our atmosphere. It isn't 21.5 something or other percent oxygen. It would be hypoxic, very, very low. I think that answered your question. But those are all the design things that I had to kind of figure out over time. Now, I almost always just put them in a single series nowadays. It's just easier. But I almost always am working within aerobic systems. So it's not an easy job, but I kind of have the easier environments to engender, I guess you could say.
So Colin, I think we have to wrap it up here because we're out of time, but thank you so much. This was a great presentation, and encourage anyone to get in touch with Colin if they want more info or to discuss this further. Thanks everybody
Thank
for
you very
coming.
much for the opportunity. Christina, thanks for getting me on board with this one. Patrick, thank you for all the connections as well. And we are looking here soon to either begin to write more papers and articles, or there had even been a little bit of talk at one point of maybe developing a journal of this kind of